The overall goal of this bioconjugation method is to fluorescently functionalize disulfide-containing virus-like particles.
This method can help answer key questions in the bioconjugation reaction for virus-like particles which have a limited number of amino acid residues that can be functionalized. The main advantage of this technique is that by using one reaction, new functional groups can be introduced and conjugation-induced fluorophores can be formed at the same time.
In addition to Qβ, we believe this reaction can be applied to any virus-like particles that possess disulfide bonds.
Prior to starting the procedure for expressing the Qβ bacteriophage, wipe down the bench area with a 1:1 bleach to ethanol solution. In an aseptic environment, make 2 3 milliliter starter cultures by adding single colonies of E. coli BL21 into SOB media. Grow the cultures in a 37 degrees Celsius and 0% relative humidity room, with shaking at 250 RPM overnight. On the following day, remove both 3 milliliter starter cultures from the shaker. And in an aseptic environment, pour each starter culture into a 2-liter baffled Erlenmeyer flask with 1 liter of fresh SOB media. Place the 2 flasks of inoculated media on a shaker at 250 RPM in the 37 degrees Celsius and 0% relative humidity room. Grow the bacteria until they reach an optical density at 600 nanometers, or OD 600 of 0.9 to 1.0. This usually takes about 5 hours. When the cultures are at the desired OD 600, use a P1000 pipe.